Hi everyone, I'm Julia Abbasi. I'm a third-year graduate student from Plant Sciences Department, and I work in Young Devojack's lab, and I work with bee genetics. I start with the student speaker. Our first student speaker is Jeffrey Neyhart. He's a fourth-year PhD student from the University of Minnesota, and he to the two-row barley breeding. So Jeffrey Neyhart.
Thank you to the organizing committee for inviting me to be here. As a organizer for the University of Minnesota Plant Science Symposium, I understand how much work and how much time goes into these events, and I'm truly grateful that you're able to host me. My name is Jeff Neyhart. I work on barley breeding and genetics at the University of Minnesota with Dr. Kevin Smith, and today I'm going to talk briefly about one of my projects in my PhD research dealing with long-term genomide selection and how we can update data in our training populations. I'll first talk in an introduction of what genomide selection is, so we're all on the same page. I'll talk about why we're using it at the University of Minnesota and the impetus behind this research, our methods, some results, and then some implications for real-life breeding. And I'll also say, if you'd like to look at these slides, they're available on this link here. And then if you're interested in following me for occasional genetics and puns, follow me on Twitter. So in genomide selection, we're using genomide markers to predict the value of individuals so we can select them without necessarily using phenotype data. So we have two different populations as demonstrated in this diagram here, a training population and selection candidates. Our training population has been phenotyped and genotyped with genome-wide markers, and we use the association between those phenotypes and the genotypes to predict marker effects. We then have a population of selection candidates that have been genotyped for genome-wide markers, but not necessarily phenotyped, and we can predict the genotypic value using that genotype data and the marker effects that we've predicted. And the advantage here of genome-wide selection is that we can make selections on the predicted genotypic values, saving time and resources and avoiding potentially costly field trials. And so we're using this method at the University of Minnesota in our new two-row spring barley breeding program. There's been a lot of push from local craft brewing industries for two-row malting barley, and so we've developed this new breeding program for Minnesota growers. However, in Minnesota, there is no spring two-row barley, so we need to improve our populations to develop adapted material and eventually varieties. And so genomide selection is a really important tool in this endeavor because it allows us to rapidly improve populations by making selections on predictions and not necessarily phenotypes. And another advantage of genomide selection, especially for barley breeding, is that we can rapidly undergo breeding cycles. And so in our program, we are able to complete a breeding cycle using genomide selection in 1 year. And this is a diagram of our breeding cycle using genomide selection. In the fall, we make a cross, we advance that population to the F3 stage where we genotype it, predict the genotypic value, and then make selections for either variety release or for parents for the next cycle. However, the ability to use genomide selection in breeding depends on the accuracy of those predictions. And this is a function of a number of different factors, including things like marker QTL linkage disequilibrium, the marker density, the heritability and genetic architecture of a quantitative trait, but then also on the design of the training population, which I'll focus my talk on today. And the design of a training population includes things like the number of individuals in a training population, the relatedness of that training population to the selection candidates, and then things like the reliability of those predictions from that training set. And so to illustrate an example, I've included a figure here from a paper by Aaron Lorenz and my advisor Kevin Smith, where they looked at barley genomide selection and building a training population of individuals that are decreasingly related to a selection set of selection candidates. So on the x-axis is the size of the training population, on the y-axis is the prediction accuracy, and over time they're building a training population using individuals that are less and less related to the selection candidates. And so what they see is that over time and increasing training population prediction accuracy increases, but at a point, it starts to decrease as individuals are added that are less related to the selection candidates. And they did this for 3 traits that are important for barley: DON, which is the concentration of a mycotoxin; FHB, which is fusarium head blight resistance; and plant height. And over time in recurrent selection for genome-wide selection, we might expect that a number of different factors are at play that are going to influence prediction accuracy. So for instance, recombination is going to break down the LD between markers and QTL. Selection and genetic drift are going to form new LD that needs to be captured. The training population is going to become less and less related to the selection candidates. And to illustrate this, I've included a figure from Javier et al. 2007 in genetics, where they looked at a training population, which is at generation 10, and then selection candidates which are at different generations. And so they see that prediction accuracy is highest when the selection candidates are at a generation that's close to the training population. And so all of this suggests that over recurrent selection, using genome-wide selection, we need to update our training data. However, and this is not a new concept and has been proposed previously, but what we wanted to look at were different methods to optimally update that training data. And so our objectives were to simulate a barley breeding program using genome-wide selection and determine optimal methods of updating training data by looking at the prediction accuracy over time, response to selection, at genetic variants, and then a number of other explanatory factors. And then I'll also suggest implications for a breeding program using the data and results that we obtained. Our starting material for our simulation included historical genotype data using SNP markers from 764 barley breeding lines from the University of Minnesota and North Dakota State University. And the relationship between those individuals is captured here on this PCA plot. I'm happy to continue the trend here with talks that include principal component analysis. But you can clearly see the difference in population structure between the University of Minnesota and North Dakota State University. These breeding programs are pretty closed, and they tend to make crosses within each program. We simulated a trait that was controlled by 100 QTL and had a heritability of 0.5. And then for each of our simulations, we extended the breeding program out to 15 cycles. This slide here demonstrates the outline of each cycle. And I know that's a lot, but I'm going to break it down. In blue are steps that we took to manage the training population. In red were steps to create the populations. And in green were steps for selection. And so in blue are steps to manage the training population. At the beginning of a cycle, we have our training population. We use the phenotypes and genotypes to predict marker effects. And then at the end of the cycle, once we've updated the training population, we use that for the next cycle. In red, at the beginning of the cycle, we simulated 50 crosses using 100 individuals. We derived 1,000 F3 selection candidates, used the genotypic data from the selection candidates and the marker effects to predict their genotypic values, and then selected on them in green. 2 different streams of selection here. For every single case, we took the top 100 selection candidates based on predicted genotypic values and used those as parents for the next cycle. We took 150 selection candidates that were selected in 7 different ways and used those to update the training set. About what are 7 methods here. For top, we took the top 150 selection candidates based on predicted genotypic values. For bottom, we just did the opposite. For tails, took the top 75 and the bottom 75 together. Random was a random subset. And then these 2 criteria, CD mean and PEV mean, were used to optimize the individuals that we added to the training population. CD mean is expected to maximize the expected reliability of predictions. It's expected to minimize the prediction error variance. And then finally, as a negative control, we did no change, so the training population remained constant. Then we also had 2 different scenarios here, cumulative and window. In cumulative, we essentially build the training population and it expands by 150 individuals each cycle. And then with window, we're using a sliding window approach where only the newest data is included and older individuals are excluded. And so the first thing we looked at was prediction accuracy. And the big headline here is that we've confirmed not updating the training population is pretty detrimental. And so on the x-axis here, I have breeding cycle, and on the y-axis is prediction accuracy, which is measured as the correlation between the predicted genotypic values and the true genotypic values of individuals. All of the different methods are in different colors and shapes, and this coloring scheme will be consistent over the next 3 slides. And so what we see, the green line, which is not updating the training population, we see this precipitous decline in prediction accuracy, which leads us to believe that our response to selection will be lower. But we also see that updating with the top method and with the tails method tends to give the highest prediction accuracy, albeit by a small margin. And we see that these 2 methods are superior in earlier breeding cycles, but that all methods, except for no change, pretty much perform similarly over the long term. And so we also looked at the response to selection here, measured as the genotypic value of the selection candidates, and then the genetic variance, which is the variance among genotypic values. At the top is the genetic variance, and the bottom panel is genotypic value. And these patterns tend to reflect prediction accuracy. When we have very low prediction accuracy, our response to selection is high, and genetic variance is quickly depleted. And then the opposite is true, which is what we'd expect given the breeder's equation and quantitative genetic theory. But what this shows is that in terms of practical breeding, since we have lower prediction accuracy for methods like the no change method, we have this selection limit where we see this earlier plateau in response to selection and total genotypic value, suggesting that we're not reaching the maximum that we could for breeding for this quantitative trait. To dissect further what was going on in the simulation, we looked at 2 other parameters, the genomic relationship between the training population and the selection candidates, measured using a kinship matrix, and then the persistence of LD phase, which is essentially the persistence of patterns of LD between markers and QTL between the training population and the selection candidates. To do that, we measured, it's essentially the correlation of R, where we take the genotypes of the QTL and the genotypes of the markers and correlate them for every pair, and do that for the training population, and then do that also for the selection candidates, and then correlate those correlations. So for the genomic relationship, we see that updating with the top method and the tails method gives us a training population that's more closely related to the selection candidates. So we're capturing those genomic relationships, we're capturing the information that's necessary to make accurate predictions. At the same time, these 2 methods also result in more consistent patterns of marker QTL LD between the training population and the selection candidates, where we see that these 2 methods give us the highest persistence of LD phase. But we also see differences between the cumulative approach and the window approach. And this might be simply explained by the fact that in the window approach, we're using only the newest data. So individuals in these training populations are more closely related to the selection candidates of the next cycle. We're getting rid of that old information that's not necessarily reflecting those genomic relationships or those patterns of marker QTL LD. And so what this means for a practical breeding program is simply, 1, we've confirmed that we need to update the training populations over time, or else we're going to see a limit to response to selection and a decrease in prediction accuracy. Number 2 is that a breeder might update their training set very simply. So a breeder is going to presumably want to evaluate individuals that are predicted to be superior to either confirm those predictions, use those individuals as parents for the next cycle, or use that information for potential variety release. And so this data that the breeder is going to generate can automatically just be placed back into the training population to help buffer against this decrease in prediction accuracy. And so this is an efficient use of resources where a breeder can simply use the information they're going to generate anyway to update their training set. And then lastly, more recent data is more informative. We see that with our window approach, where we're only including more recent data, this information better captures genomic relationships between the training set and the selection candidates, and then also captures marker QTL LD information. And so I'd like to acknowledge everybody that helped me with this research, including my advisor, Kevin Smith, and everyone in the Smith Lab, past and present. My co-authors, Aaron Lorenz and Tyler Teedy. Contributors, Ahmad Salam and Celeste Falcon, who helped with earlier discussions for this research. Resources from the Minnesota Supercomputer Institute, and funding from USDA, the U.S. Wheat and Barley Scab Initiative, Minnesota Department of Ag, and RAR Malting Company. And then before I take questions, I'll need to plug that this research was accepted at G3 and is available in the early online edition. And then you can also access all of the data from our lab's GitHub repository. And with that, I'd love to take any questions. Thank you.
So yeah, any questions for Jeff? Okay.
How sensitive are the results that you presented to differing genetic architectures, so different numbers of QTL or frequencies of QTL?
We didn't explicitly look at those differences, but we'd expect that a trait that is more highly heritable would result in more accurate predictions. I don't know if there would be an interaction between genetic architecture and the patterns that we saw between methods. So I can't say anything for sure, but I would expect on general that lower heritability would result in lower accuracy and higher heritability with higher accuracy.
So did I miss it, or did you not present any changes in the genetic variants in the breeding population over time? And did you look at that for the different methods?
One of the slides did show change in genetic variance, which was the variance among genotypic values. So that tends to decrease as selections are made. Even with more accurate selections, we're accurately capturing the effect of QTL. And so when we make selections on that, we expect that the frequency of those QTL would decrease, or more favorable QTL alleles would go to fixation. And so given that, we see a decrease in total genetic variance. All right, thank you.
Okay, thank you, Jeff.